assumption.
Professors
Eric
get
said we
vacation
could
do
go on
that.
vacation.
Okay, so what we're going to do today is we're going to use a Powered by iPlant program. And this is, like I said before, one that my group maintains called CoGe. And CoGe, just to give a brief history of the project, what we were really focused in on was comparative genomics. And when I set about designing this platform, I just sort of made the realization that no matter where we were today, tomorrow we were going to have new versions of every single genome and then 10 times many new ones coming online. So when this platform launched, we had, when I first started it, we had 2 genomes, rice and Arabidopsis. And now today, based on good scalable technology behind the scenes, a lot of it is based on engineering and a lot of it is based on what iPlant helps us with. We've scaled to well over 20,000 genomes from all domains of life. And so over the last year, one of the big pushes that we have been doing with this platform, so this lets you do all sorts of fun stuff with genome analysis. But the big need that we were finding was to start incorporating into it what we'll call quantitative genomic data, so the ability to do measurements and associate that to a genome. These are things such as RNA-seq, methylation patterns, copy number variations, so on and so forth. And then we have a bunch of other kinds of data associated with the genome, specifically around diversity. So SNPs, variants, all those kind of good things. So we've been working on the underlying technology so that in the same way that we can store thousands of genomes, we can store thousands of what we call functional diversity experiments per genome. And so we have in place ways for people to add in their data, mix and match it with public data, keep it private, share it with other users, and if it's made fully public, anybody can use it anonymously. So we try as much as we can to make it easy for people to integrate, share, and publish their data. Underlying this, again, is iPlant. And one of the key technologies that we've leveraged of iPlants is their user authentication system. So what we're going to do is a bunch of hands-on. This will be very quick in terms of basically using CoGe to map some RNA-seq data to a genome in the system, again, using Arabidopsis. So CoGe's homepage, genomeevolution.org with one E between genome and evolution. Use Chrome, Firefox, anything but Explorer. And even then, we do have some problems with some of these browsers. So if you run into any difficulties, let us know. At some point, you may get a pop-up blocker. Go ahead and just enable that if you do see the pop-up blocker pop up. And once everybody's at genomeevolution.org, just raise your hand. Give me a visual cue to let me know that you're all in the same spot. Of course, the Slacker's up here on your front left. We don't know what they're doing.
I'm having a hard time. There's only one E.
Genome with 2 E's should work. I just can't remember if I've kept that domain address up to date.
It come up, and then I tried to start Firefox and see if it would come up there. And
Yeah, it's just
Have you tried Chrome?
yeah, I went through Chrome, and then you said Firefox.
The fun is
Chrome. Use Chrome.
Alright.
We mostly do our testing in Chrome.
That's what she's in right now. It's just not letting come in. Are you connected to the
Aspergillus oryzae, one of the few non-toxic forms.
I was using Discovery Garden.
Yeah.
Very good.
Laurel,
Yeah?
these kinds of complicated problems I don't know if we can help with.
I don't know.
It's just not loading.
Try to go to Google
Yeah,
and
right.
type search for CoGe at Google.
Right.
Yeah, that's what
And
I
you
was
can
using the genomics.
Yeah, this is how to use this with that.
So, and Laurel, if you do need a hands-on later, I can definitely walk you through this.
Okay, Google's kind of irritated with
So-
me right now too.
So if Google's irritated with you, then it's your internet connection. So, once you come to CoGe, you have access to a whole bunch of fun stuff, but I want you to log into the system using your iPlant credentials. So if you click in sign in the top right, this will actually bump you just straight over to iPlant's authentication system. Type in your iPlant username and password, and And then iPlant is going to kick you back over to CoGe with a very special, if people are interested, short living token that CoGe will then use to do a second round of authentication on you. And if you pass, it will let you into the system, and you'll see your name in the top right. And if you don't pass,
You're not you.
just don't take it personally. So again, there's a lot to CoGe that I'm not going to cover today. We're just going to focus on doing RNA-seq and integrating it with the genome. So what I want you to do is click on the My Profile button in the top right corner. And what this is basically the place where you can manage and share and see data that has been shared with you. And so when you log into My Profile, in my account, this is my real account. I've got tons and tons and tons of data in here. Things with the little restricted mark, those are private data. So this particular genome, which is a bird, is restricted data at this point, to which I have access. Over here, you have who you are. You can click on that picture and change your icon, personalize it any way that you want to. This is purely for your amusement. And then what you have over here on the left is a bunch of ways that you can sub-select out of your data. So you have My Data. This is stuff that you've added. Notebooks. These are collections of data that you've generated. User groups. This helps make management of who has access to what a little easier. So in the case of these bird data, there's a big consortium that has sequenced 50 bird genomes, and I'm sort of the guy that's responsible for managing those data. So I load them in there, and then what I do is basically make sure that the right people have access to it. They all get iPlant user accounts. I take all these put them into a special user group called Avian, and then grant that entire group access to all of these genomes. If it's got a double helix, it's a genome, and if it's got a flask, it's an experiment. There may be some experiments in there, there's some experiments. Shared with me, these are data sets that people have shared with you. And if you start using the system, it records every analysis that you do and generates a unique URL that you can use to revisit in the future. And at any point, if you want to, you're welcome to delete your data. This actually doesn't delete it out of the system, but it just marks it in a way that it doesn't show up anymore for anyone. And if you ever want to, you can go ahead and delete it. So, quick overview of how this data management system works. If you do have data and you want to share it with someone, click on the person icon. This here lets you add it into notebooks, and this here trashes it. And this here lets you send items somewhere. So in this case, what we're going to do is we're going to load in new data. So if you click on the Create button, this is where you can create new user groups, new notebooks. You can load your own genome into the platform, keep it private, again, share it with collaborators, add in structural annotations and functional annotations. But today, we're going to add a new experiment. So all of these functional diversity data we consider to be an experiment. And so what we're going to do is we're going to go get some example data and map it to this genome. And so I'm going to go and show you where to get these example data from. So what it's done right here is it's opened this up inside of your user profile page, but we're just going to actually we can just say open up in a new tab, and it's going to take you to the parent tool that does this, which we call load experiment. You guys all with me so far? All right.
So you're saying that the
Was
experiment
it our
is
fault or your
anything that isn't a genome?
Yeah, any functional diversity data to which you associate to a genome. So resequence data is taking reads from an individual and then mapping them to a reference genome. That's the reference, and then everything else is sort of an experiment that we've done against the reference. Basically quantitative, what we call quantitative data, so some numerical measurement, or state data. So at this position, instead of a T, there's an A there, or a small deletion, or something like that. Yeah. So once you're in load experiment, if you notice at the top right, we have the exact same menu as you saw before. One of them is called help. And so if you ever play with CoGe, help is a good place to go find some help. But click on the link called Page Docs. And what page docs is going to do is take you to our internal wiki system that we use for writing tutorials, documenting the system, putting in some neat analyses that we see. And what this does is it basically walks you through all the information that you need about how to load data into, experimental data into CoGe. So if you were to load in, so let's say, for example, you were using the discovery environment, and then inside the discovery environment, you generated a BAM file or a VCF file. You then can use that file and integrate it within CoGe, and then once it's integrated in CoGe, you can use our genome browsing system in order to visualize these data on top of your reference genome. But the thing that we're interested in is FASTQ data. And so down here at the bottom are some example data sets. If you follow this link, we have some pre-processed data that you could add in there if you really wanted to get into the system. A genome, some structural annotations, expression data, variant data, and then the BAM file. But today, again, abbreviated, focusing in on RNA-seq. So here's some RNA-seq data that we're going to be associating to an Arabidopsis, just like in the other example. And down here we have 3 different densities of data. One with 170,000 reads, one with about 1 million reads, and then one with about 7.5 million reads. Because I want you all to run this, and I want this to actually complete in a very short period of time, we're going to go for this small data set, the 0.17 million reads. All I need you to do is copy this link. And you can do that 2 ways. You can select it and copy, or you can right-click on it if you have that enabled and know how to do it.
Copy link.
And then copy link address.
Sorry.
So now, I'm just going to wait a moment just to make sure everyone gets a chance to copy that link address.
How do I go to example data?
Sample data. Go to load experiment, go to the help. Under help, there's page docs. If you click on the page docs, it's going to open up the documentation for load experiment. And if you look at the table of contents for load experiment, at the bottom are example data sets. Click on that, and that'll take you right down to those data sets. And if I went too fast, I can do it one more time. Or we'll have one of these folks help out. So now we're going to load your experiment. Give your experiment a name. I'm going to call this one test, in class, live, dangerous, live demos. Version 1, source. Source is where you got these data. We make it a requirement that you attribute these data to somewhere, someone. If you go ahead and search, for example, if I search for the word test, there's a bunch of things with the test in there. If I need to create a new data source, I can go ahead and click on new. And this gives you a chance to add a name, description, and link. So if it's out of your lab, and your lab's not listed, or some other source out there. So here, I'll go ahead and say that this came from iPlant. iPlantcollaborative.org, why not? Restricted, whether or not these data are going to be made public. Do not make these data public. Genome, now we're going to have some fun. We've got to go search for Arabidopsis thaliane and a specific version of it out of 20,000 genomes inside the system. So you can just start typing in an organism name, but the fastest way to find it is going to be type in col-0 or Columbia-0, which is the typical genome. Give it a moment. It's now going to go hit the database, and we're probably all hitting it at about the same time. And it's going to come back with a list of genomes to select from. I want you to select this one. This is, so somebody's made, we let people make copies of genomes, and somebody made a copy of Arabidopsis and made it public. I have no idea what that is. But the genome I want you to look for is the one that has the ID of 16911 in the description. And I'll leave that up and wait for everybody to come to speed. And if you want to, go ahead and click Load Experiment. And what this will now do, behind the scenes, is it will launch CoGe's RNA-seq processing pipeline. So what we did for this pipeline is all of us on the team basically said, you know what? I don't know diddly or squat about really how to do RNA-seq as really well. But I know that there's people who are great out there doing this. Do you have a question?
Send to select data.
Yeah.
Skip to select
That'd be-
data.
I forgot to let you guys add in your data.
Yeah.
Details. Details. Okay. So add data files. Select data files. So CoGe lets you pull data from a variety of places. You can upload it from your desktop. What it's doing is it's going into a special directory inside your data store under the iPlant data store tab, specifically a directory called CoGe_data. And it'll let you pull data out of your directory. So if you do have a 30 gig file, you just pull it off the sequencing machine, put it in your iPlant data store, put it in the CoGe_directory, and then CoGe can go and pull those data for you automatically. This makes our data management easy because you now can leverage iPlant for managing gigantic gigs of data. And we don't have to. And we can't. The technology is complicated to stand up. But in this case, what we're going to do is we're just going to retrieve it from an FTP site. So if you do have somebody has posted your data on the web, you're going to download it from the Genome Center. They've given you a URL. You can go ahead and just paste in that URL. In this case, this is data that's in the iPlant data store. And then just get. Press get, and then CoGe will go get your data for you and load into the system. This does have support for simple HTTP passwords. So if it is under a simply protected site, my meeting is in 10 minutes. You can go ahead and do it. So go ahead and add that file. It's uncompressed. It's about 45 megs.
Do you want to use Tomcat?
Use GSnap. GSnap is better and faster. It's not as popular.
So all that stuff we learned for the other hour and a half.
Right.
Forget that.
No.
It's an
So
alternate way of thinking about it.
Yeah.
it's a different mapper. All the other steps are going to be using the Tuxedo Suite tools. So once that's in, now hit load experiment. And then we'll now launch the pipeline. So while this is running, I want you to go back to this help page.
I think we all got failed.
Yeah.
I just got rid of mine, but it said it failed.
Yeah, I got
Yeah.
it failed.
Try that.
Dangerous.
It says failed.
Maybe we were all trying to do it the exact same
Maybe
time.
it was.
Israel, fine. So who's someone that failed?
I hit okay, but everybody failed it over here.
Windows submission failed. Hit OK.
Yep, everybody over here too.
It's still trying to load the experiment.
I think
But I hit okay.
Eric, I think you're the only one that's allowed to do it.
Did yours work?
Yeah.
And you're on a Mac. Everyone else has a Windows machine.
So that's the true.
Huh.
I do Linux.
Oh,
yeah.
Jerry thinks it works. Apparently you have to be an iPlant person. No.
No, but you guys all got to fail. Let me pick someone's computer to do it. Can I try yours?
So what is the
Out of
version?
that.
The version is just put in one.
Yeah.
Doesn't matter.
I mean, like when we're using this for ourselves, what will that do? Will that add like?
Well, if this is your own genome, my assumption would be that you would have a version of that genome that would be internal to your own process.
Right.
So-
But when we run this, is it?
Did you
just
select a valid data source?
It tells you
I
what
put the
version
iPlant.
in your mind.
Right.
Okay. Because they released new
Totally
versions.
new.
I've got an idea of what's going on.
Yeah.
Maybe.
But I think that, to me, it would be intuitive to say, this is my experiment, this is my version of this. Oh, I might go do it again. So that way you see it, it'll version it for you.
Right. I mean, I've been running like highest data, the genome transcriptome to get some of the lines that I've sequenced in the last 6 weeks. Another.
I believe so. I haven't
Trying to filter that.
delved into anything else.
This is for your, since this is your